Yeah, so we're going to be talking about biochar, and that's a sustainable byproduct of agricultural waste that's created. And we've used it for research for a number of purposes and for basically killing or removing Salmonella and E. coli from soil, compost, poultry litter, and irrigation water. But I did want to first start out just talking a little bit about our organization, the USDA Agricultural Research Service, or ARS. The ARS is one of about 15 different arms of the USDA. We're 100% research, and our positions are comparable to a university research professor with a 100% research appointment. Our research
Joshua, you're frozen, unfortunately.
research projects, 2,000 scientists and postdocs, and 6,000 other employees in 90 locations in the US as well as overseas. And we have a budget of $1.7 billion per year. We are just outside Philadelphia. We're actually on the edge. Our property borders the Philly city limits. And we are the Eastern Regional Research Center in Wyndmoor, Pennsylvania. And we were established in 1940. Our center was built in 1939. This is a shot in 1947. You can see it was basically farmland in the back in 1947. That was 1960. You can see it's getting built up. And then there is modern day. When the center was built, there were three other centers built: Western, Northern, and Southern. And we are the Eastern. They all look exactly the same. They have this horseshoe shape. This is one of our pilot plants, and they have developed a number of agricultural and food products there. We have been remodeled in the past couple of decades. We had a couple of remodeling. Those are our labs. This is actually my lab there. One of the products that's been developed here where we're at in Wyndmoor is based on the potato farmers after World War II had no place to sell their potatoes. And so for centuries, at least a couple centuries, they had been trying to develop a dehydrated potato that could be rehydrated into mashed potatoes. But unfortunately, when they cooked the potatoes, it would break the cell wall. And so when they rehydrated the mashed potatoes, they would turn into glue. Well, in the 1950s, at the ERRC where we're at, they developed the potato flaking process, in which they learned to cook the potatoes without breaking the cell wall, dehydrate them, and create the products that you see here. Potato flakes, which has been a multi-billion-dollar industry. They also developed the low-fat mozzarella cheese that's used in the U.S. school lunch program. They developed lactate milk in the 1980s. They conducted the work that went into the approval for irradiation of meat and leafy greens. And there's our pilot plant. If you look in the very bottom, there's a machine. That's the vacuum steam machine. And this was based on a problem with hot dogs and frankfurters and sausages that have listeria in them post-processing. And the vacuum steam vacuum process that they developed was commercialized and marketed by Johnsonville sausages to kill salmonella in packaging. We also have a very active biodiesel program, which is how I got into biochar that we'll talk about. A very strong residue chemistry program, and a number of different technologies that we work with: UV light, gamma radiation, high pressure, gaseous decontamination, pulse electric field, cold plasma, laser treatment, infrared, ultrasonic, supercritical CO2, flash pasteurization, antimicrobial packaging, and nanotechnology. I've worked with a number of those, but the main two I've worked with are antimicrobial washes for fresh produce, as well as biochar application to soil. This is a snapshot of my lab with some of my former grad students and employees. The first thing I did when I was hired 17 years ago is I was tasked to work on liquid egg pasteurization. 3 billion pounds of liquid egg are pasteurized every year, and these go into products such as scrambled eggs, sauces, salad dressings, etc. If you ever eat at any major chain restaurant and eat scrambled eggs, they don't break the eggs there. They actually use these pasteurized eggs they receive in bags. So we performed experiments for a few years on this, and this went into writing the September 2020 FSIS Food Safety Guideline for egg products. We also did a number of studies with pulse electric field, pasteurization of orange juice, and strawberry puree. We did some leek and romaine lettuce root internalization studies to determine if Salmonella and E. coli are internalized into the plant through the roots based on arbuscular mycorrhizal fungi that's in the soil. I had a postdoc for a few years that did some norovirus work with surrogates. And then we conducted a series of studies inactivating Salmonella and E. coli on tomatoes and strawberries. This was in the wake of the strawberry outbreak with E. coli that made a number of people sick. And in this, we developed a process where you draw a vacuum when it's being washed and then release the vacuum, and that allows the antimicrobials in the water to infiltrate the pores. This is an SEM pick, and this is actually the stem scar where a lot of bacteria is internalized in the tomato. What we did is we inoculated the outside of the stem scar, but then we cut it out and sectioned the inside. And you can't see here, but the bacteria actually infiltrated down into the stem scar, as you can see here, and went deep into the stem scar. And so we developed a number of antimicrobial washes. We tested scores of different combinations of antimicrobial compounds. And from that, we collaborated with the company Nature Seal out of Rhode Island, and we developed a patented organic produce wash called, it was marketed as First Step Plus 10. I developed another conventional produce wash that was just patented in June of this year. We also conducted some Salmonella apple dehydration studies and published a couple of papers based on some industry needs of E. coli and Salmonella surviving the dehydration process. As Amy mentioned, I'm the editor for Journal of Food Protection, and if anyone is interested in reviewing, if you are have a PhD or you are in grad school to work on your doctorate, let me know if you would like to review for the journal, be an ad hoc reviewer, and possibly be added to the editorial board. So for the remainder of the talk, we'll discuss biochar. So the reason I got into biochar is because there's a significant problem with pathogen contamination in crop soil. This largely results from farm animals, livestock, and poultry, as well as wild animals. And there's a significant problem with manure disposal in the U.S. I published a review article on composting to inactivate foodborne pathogens in crop soil that covers a background on a lot of this. Now, in 2004, this was 20 years ago, there were 1.3 billion tons of manure produced a year. I don't know what the current amount is, but conservatively speaking, let's say it's 1.5 billion 20 years later. That's 3 trillion pounds of manure that's produced in the U.S. every year. That largely contributes to a lot of the contamination problems in fresh produce from water, irrigation water, and runoff. One study reported that Shiga toxin E. coli could survive in manure for over a year. So it is a significant problem. Therefore, a number of studies have recently been looking at decontaminating crop soil because some fields have to be taken out of production if they test positive for these pathogens. I published a review on this on pathogen decontamination or remediation of food crop soil that covers this in detail. We'll discuss this just briefly. Although decontamination of crop soil has been conducted for decades, its main purpose has been the elimination of phytopathogens or plant pathogens. However, there may be an overlap in application. In the past, little was published on evaluating an activation of human pathogens in crop soil until probably the past decade. There have been a number of labs that have started working on this, some actually out of Maryland, the ARS there, North Carolina State, and us in Wyndmoor, as well as maybe Kansas State, have been looking at ways to inactivate it in the soil. And so some of the ways that are used to inactivate plant pathogens in soil may also be applicable for foodborne pathogens. Some of these methods used to inactivate plant pathogens include soil solarization, in which they cover the field with tarps. And with black tarps, they've reported temperatures over 70 C, surface and subsurface steaming that increases the soil temperature up to 71 C, soil fumigation. This is actually an ARS picture of some research that we did on soil fumigation. Now, the problem with soil fumigation is that methyl bromide is the main chemical that has been used for decades, but it was phased out in 2017. So they're looking for alternative fumigants. Antimicrobial drip irrigation or chemigation, parasitic acid is often used. Biofumigation, in which you grow crops that contain glucosinolates. These release antimicrobial isothiocyanates from the crops that kill the bacteria. Soil flaming. I have a background in the poultry industry, and when I was in poultry, there's a company that sells equipment to flame the soil and flame the poultry litter. That can be used. And the one we're going to look at is biochar application. Well, first of all, what is biochar? Biochar is black carbon generated from pyrolysis. It results in incomplete combustion. The temperatures are often between 350 and 750 C. And this is conducted under low oxygen or no oxygen conditions. Some of the major biofeedstocks that are used are listed here in this chart. Actually, the major biochar biofeedstock used in the world is actually animal manure, dried animal manure. But other biofeedstocks, the major ones used worldwide, are coconut shell, rice husks, wood, and various types of crop residue. Now, to show you, if you're unfamiliar with biochar and biochar research, this shows you the significance and how fast it's growing. A May 2024 Scopus search that I conducted revealed that in the past 20 years, there have been over 24,000 publications about biochar around the world. Over half of these are from the People's Republic of China. We have a very active biochar program within the Ag Research Service of the USDA, and a dozen or so scientists working on that around the country. We have a process in which it's thermochemical conversion. It's called fast pyrolysis to produce biochar. And you see this unit here, we call it the CRIPS unit, and it's based on this fast pyrolysis process. In this process, the biomass, let's say corn stover or sorghum bagasse or wheat straw, is ground into a fine powder. It's run through this sand furnace. Let me see if I can, it's run through this sand furnace, and this is a pyrolyzed bed that's heated up to 500 or 600 C. This separates into three components. It condenses into a bio oil, and secondly, it creates a syngas. This syngas is fed back into the system to make it self-propagating. The syngas is largely composed of methane. And then finally, there is a biochar, which is what we are actually interested in. This CRIPS unit that we have can produce up to 40 gallons of biofuel a day. It also produces electricity. Now, these are some pictures of biochar as they're applied around the world. They're mainly applied to soil. Here are some pictures where they're applied via hand or tractor or injected into the soil. This is in Hawaii. They do a lot of biochar in Hawaii. And the benefits of biochar to the soil is it provides nutrients, it increases the crop yield in poor soils. What you see to the right here is biochar in soil, and actually in the terra preta soils of the Amazonian dark earth in South America. The natives over 2,000 years ago, according to archaeologists and anthropologists, they tell us that they practice slash and burn crop type production, in which they slashed and burned byproducts and woody waste in order to produce this biochar in the soil. And this is still viable in there for over 2,000 years. It also locks up the carbon for up to 2,000 years. A recent publication reviewed the benefits of biochar. And in this meta-analysis, they looked at 57 published articles and noticed that using biochar increased crop yield by 25%, but using, organic fertilizer increases it up to 22%. However, when you combine inorganic fertilizer, manure, along with biochar, it increased the crop yield up to 180%. For an example, this is in Hawaii. These are green beans without biochar on the left, and green beans with biochar on the right. Bok choy without on the left and with biochar on the right. Well, what are the benefits, other benefits of biochar to soil? It increases bacterial diversity. It's also used, lots of studies use it to bind heavy metals as well as chemical contaminants. It reduces carbon gas emissions as serving as a carbon sequestration sink, and it remediates poorly drained soils and soil with a high clay content. So for the remainder of the talk, I will discuss our biochar studies at the ERRC. So the first study we're going to look at, first studies, are biochar added to soil and compost to decontaminate it from E. coli and or Salmonella. The first study we conducted some years ago was looking at the inactivation of E. coli 0157H7 in soil with fast and slow pyrolysis biochars. And this began because I attended a seminar on biochar by one of our researchers, who's now a research leader at the center. And I asked him, do you know if this will kill bacteria in soil? He said, I don't know, but I'll give you some if you want to try it out. So he gave me a number of different types. We published a paper on this. And now I'll give you the background of the study. We screened 12 types of biochar to inactivate E. coli, and we added it at a very high level, 10%. This is higher than you would add in soil. But we wanted to add it at a high level just to see a baseline pilot study to see if it would inactivate it. We looked at soil over an 8-week period with 18% moisture at 21C. These are the different biochars we looked at. The top 4 are fast pyrolysis. That goes through the fluidized bed for less than 1 second. And the bottom ones are the standard slow pyrolysis, where you just cook it at high temperatures for in between 30 minutes and 2 hours. We looked at switchgrass, oak, horse litter, and hardwood pellets. The slow pyrolysis biochar was generated with the Biochar Experimenters Kit. This used to be marketed by All Power Labs out of Berkeley, California. I'll mention them later. But these are our results. Notice at the very top, you'll see the top line is the soil positive control. This is the soil with the E. coli without any biochar. Now notice at 4 weeks, you still had about 5.8 logs of E. coli, but drop down to where it says FP switchgrass. With fast pyrolysis switchgrass, we inactivated 6 logs within 4 weeks. So that was good, but if you look at the right, notice for the soil positive control, this bacteria at 8 weeks totally died off without any biochar. So it may have been weaker isolates or strains of bacteria. So our next study, we actually looked at a number of different types of bacteria. We looked at foodborne pathogens as well as non-pathogenic surrogate organisms to see how it was affected by biochar. So the objective was to determine the influence of 7.5% biochar. We lowered the percentage of biochar on 7 different, not bacteria, but 7 different bacterial composites. Each of these composites contained 2 or more isolates. And so there were a total of 33 different strains we used. We actually tested this in dry soil, 7% moisture over 90 days at 21C. Here are the bacterial composites we looked at. For the pathogens, we used 5 strains of O157H7, 6-strain composite of Salmonella, and a 6-strain composite of non-O157 Shigatoxin producing E. coli. Now, here are the non-pathogenic bacteria we use. These are potential surrogates. We used a 7-strain composite of non-pathogenic E. coli from ATCC. Then we used 3 strains of attenuated E. coli O157H7. This was from Mike Doyle at University of Georgia. His postdoc removed the Shigatoxin genes from these 3 bacteria. Then we looked at 2 non-toxigenic O157H7s from ATCC. These are available at ATCC, and they are O157H7. They just don't have the Shigatoxin genes. Finally, we acquired some bacteria from Roy Curtis. He was at University of Arizona at the time. He's now at University of Florida, and he does a lot of vaccine work. These were actually 4 poultry vaccine strains that were non-pathogenic. So here are our results. This is the first graph figure is just soil. Notice just in soil, the O157H7 E. coli and the Salmonella survived quite well up to 90 days, surviving up to 4 logs up to 90 days. But notice that the non-O157H7 STAC dropped off rather rapidly. Next, we'll add in the University of Georgia Doyle attenuated O157. It did not survive quite as well. But notice this, the ATCC attenuated O157. This is the one that had the Shigatoxin genes removed. This survived quite well. And so it looked like a good potential surrogate. And so we actually use that going forward in other studies. All the other ones did not survive quite as well. Now we'll look at the biochar in the soil. When we added the bacteria to the biochar in the soil, these are the ones that did not survive quite as well. However, the O157 E. coli that's pathogenic, it kind of bounced around. The Salmonella survived quite well. And again, the purple is the ATCC strain of O157H7. We use that in all studies going forward. It looks like a good surrogate. So in the next study, we again looked at fast and slow pyrolysis biochar against E. coli in soil. We published this in the Journal of Food Protection. And we were going to look at 3 fast pyrolysis biochars as well as 10 from the UK Biochar Research Center. These are slow pyrolysis biochars. We'll talk more about that in a minute. These were all added at 10%. And we use the ATCC non-toxigenic E. coli in soil at about 17% moisture over 7 weeks at 21 degrees C. Now, this is why we use the UK Biochar Research Center biochars. So there are literally hundreds, if not thousands, of different types of designer biochars around the world. And the problem is when these 20,000 papers are published, 24,000 papers, it's like comparing apples to oranges because you have lots of different biochars and it's hard to compare them. So the UK BRC developed a set of 10 standard biochars that could be used, and they've been used in dozens of studies around the world so people can compare their results on a level playing field. So the ones we used were softwood pellets, wheat straw pellets, miscanthus straw pellets, oilseed rape straw pellets, and rice husk, each processed at either 550 or 700 degrees C for a total of 10 different biochars. Now, these are the results. When we looked at these UK BRC biochars, notice that at week 2, we stopped at week 2 because there was no significant reduction. And if you're adding at 10% and there's no reduction at 2 weeks, there's really no reason to go any further. So the UK biochars didn't do very well, but we found pretty good results with our 600C fast pyrolysis biochar. We stored this at 22 degrees C, and you'll notice that on the left-hand side, you'll see the percent of biochar that was added. When 0% biochar was added, at 7 weeks, we had 6.3 logs of E. coli still viable. But notice for the 3.5% biochar, at 7 weeks, it killed all 7.5 logs of this biochar. So that was very good results. In fact, if you look just at 3 weeks, it already dropped it down by about 7 logs in just 3 weeks. So good results. So the next study, we're actually publishing this now. We're looking at the inactivation of 0157H7 in fresh dairy manure compost by alkaline slow pyrolysis walnut biochar. We actually reported this in 2020 at IAFP, and we're only now getting around to publishing it. This is the publication. We're sending the next revision off this week. Hopefully it will get published. So our objectives were to test 2 switchgrass fast pyrolysis biochars, in addition to 2 walnut hull slow pyrolysis biochars to kill E. coli in dairy compost at 22C for 7 weeks. And we also I pointed out earlier the Biochar Experimenters Kit. So this is again from All Power Labs out of Berkeley, California. Well, they actually produced a larger unit. This is a gasifier that produces biochar. It's a 25 kilowatt power pallet gasifier, and it produces biochar as a byproduct, but its main purpose is to generate electricity. It's very similar to what we showed you before in that it creates a syngas. The syngas, largely composed of methane, is used there we go. Now I can use my arrow. Can you guys see the arrow now? I hope you can see the arrow. So,
Yes.
okay, good. So, This is a self-propagating system. You put the bio feedstock in there, it produces electricity, but it also uses this syngas to run the system, to continually run it. So it produces lots of electricity, and it does it for the cost, less than the cost of diesel, so it can be used on farms to generate electricity. And as you can see at the bottom, a fuel cost comparison, that producing electricity with diesel or gasoline costs up to 75 cents per kilowatt hour, but only up to 6 cents a kilowatt hour for using the gasifier. Now, if you compare this to electric grid costs around the country, in Hawaii, the electric grid cost just for electricity is 44 cents per kilowatt hour. In North Dakota, where the lowest cost of electricity is, it's 11 cents per kilowatt hour. But using this gasifier, as you can see here, only costs 6 cents a kilowatt hour. So it's much more efficient. They also have a much larger one, 130 kilowatt and 150 kilowatt biochar producing gasifier. Well, here were our results. Notice at the top line, this is for when we added the biochar to compost, dairy compost. On the very top line, you'll see that we inoculated at 8 logs, and after 6 weeks, the E. coli had actually grown to 9 logs, over 9 logs. Now, when we used a high temp walnut biochar, at 4 weeks, it grew to 9 logs. Look at the very bottom. For our fast pyrolysis biochars at 500, 600 degrees, both of these also grew to over 9 logs. But then notice we used this walnut cyclone biochar from the All Power Labs gasifier, and it killed all the bacteria. It killed 8 logs in only 7 days' time. Very effective at killing the bacteria in soil. That's compost. We also tested in soil, and here we added at a lower concentration, only 3.5%. And notice at 3.5%, it killed 6.7 logs in only 14 days here at the bottom. Very, very effective. Now, why was this? Well, we believe that it's largely related to the pH. This is a very alkaline, caustic substrate. And when we added 10% of the biochar to compost, it raised the pH to 10.8. And when we added 3.5% to soil, it raised the pH to 10.3. The next 2 studies were looking at the minimum concentrations of this same alkaline walnut biochar, as well as hardwood and paper biochars to inactivate E. coli. Why paper? Well, we were looking for the most alkaline biochar, and paper biochar is some of the highest pH biochar, largely because the paper process uses a lot of calcium carbonate. And this produces a high pH. Is someone talking?
Well, we're good. I think it was just background.
Oh, okay. So we published this earlier this year, the minimum concentrations of slow pyrolysis paper and walnut whole cyclone biochar to activate E. coli in soil. And we tested 31 different biochars. We looked at mainly 2 biochars, a slow pyrolysis walnut hull and a paper biochar. The significance of paper biochar also is that there are over 82 million tons of paper sludge produced a year. Most of this is dumped into landfills where it degrades and produces carbon dioxide added to the atmosphere. Here's the 29 other biochars. They did not do quite as well as the 2 we're focusing on. Here are our results. When we looked at soil amended with the paper walnut hull biochar, we looked at concentrations in between 1 and 6.5%. The black circles represent the represent the soil without biochar. Notice it only dropped about 1 log in 6 weeks. However, when we added 2% paper biochar, we started getting reductions, some complete reductions. These blue circles are the results of 3 reps. These are data points from 3 reps. So we did get a good reduction. 2% walnut hull, not quite as good. Now notice when we increase it to 2.5%, we got almost complete reduction. So 2.5% either paper or walnut hull biochar completely inactivated Salmonella and E. coli in soil. Next, we looked at Salmonella. First was E. coli, second was Salmonella. The results were even better at 1.5%. It only took 1.5% paper biochar to start seeing a reduction. A little bit less reduction with walnut hull. But notice at 2%, it only took 2% paper biochar to completely inactivate the Salmonella. However, it took more, it took 4.5% of the walnut hull biochar. Our ongoing studies that we're currently looking at are looking at minimum concentrations of paper biochar to inactivate Salmonella in poultry litter. Our preliminary results show it requires about 5.5%. And at 6%, we can get a 5 log reduction in 24 hours. And the final study I'll focus on is what we've been working on this year. This is biochar water filter, irrigation water filter studies. We're trying to remove E. coli from irrigation water. We've been working, many of you will know Dr. Manan Sharma and his postdoc Alan from Beltsville, USDA ARS. They've been doing irrigation water filter studies, so they helped us creating these irrigation water filters. So we've been testing lots of different types of filters, or excuse me, different types of biochar. We're trying to maximize the reduction of E. coli based on the type of biochar. So first we used this small biochar filter. We weren't getting very good results, so my technician said, why don't we double the size? So there's our current 8.5-inch water filter. It's a sand biochar filter. Now, our original studies, we only got between 0 and 1.4 log reduction in irrigation water. Our best reduction, and this is our unit that we now produce the paper biochar in, our best results was with paper biochar with a 50-50 biochar sand filter. You can see that's what the paper biochar looks like before we grind it on the right. Now, one area we want to look at is biochar with greater surface area. So the surface area of biochar is often between 8 and 132 square meters per gram. However, we delved into the literature and we found that the biochar can be activated, similar to activated carbon. And when you activate it, you can make it 25 times higher, greater in surface area. In fact, one study here in 2016 showed a surface area of 3,200 square meters. So in 1 gram of biochar, you get 3,200 square meters. That's about 0.8 acres or 8 high school basketball gymnasiums in 1 gram. So we're going to try to look at more of these high surface area biochars. These are just preliminary results. And these are the different biochars on the left. And I want to point out one thing here. I'm calling this brand X because we don't want to name the brand before we actually publish it. This brand X biochar, when we added it, we dried it and we sieved it and created the biochar filter. We only got 1.5 to 2 log reduction. But when we added it straight from the bag when it was still wet, we got 3.5 to 4.5 log reduction. And what we found is that when we were drying the biochar and creating the filters dry, when we run the water through the filter, it creates air pockets and it doesn't get completely moist. So we started adding moisture back after drying it. And these are our most recent results in our 8.5-inch filters. We're getting over 4 log reduction with a 50/50 biochar sand filter. And this is when we add 64% moisture to the biochar filter before running water through it. So those are good results. A 4-log reduction is very good. We're still working with Dr. Sharma Manan from Beltsville ARS, and we want to combine our biochar filter with their ZVI filters. If any of you guys are familiar with their work, they use zero valent iron. This is a byproduct of the steel industry that basically spits out electrons and can kill bacteria. The other best biochar we saw was this miscanthus straw with 72% moisture. So that concludes my talk. Thank you for your kind attention, and I'll be happy to take any questions.